A strand of DNA is made up of a sugar phosphate backbone that the nitrogenous base pairs adenine and thymine and guanine and cytosine are individually bonded to. These base pairs are bonded together in the middle of the double helix by hydrogen bonds. Adenine always pairs with thymine and guanine always pairs with cytosine.
Helicase, represented by the black triangles, is an enzyme that unwinds the DNA double helix and unzips the nitrogenous base pairs. This creates the replication bubble and provides the template strand of DNA needed by the DNA polymerase to duplicate DNA. Polymerase reads the DNA strand from 3' to 5' and builds a new strand from 5' to 3'. Polymerase begins building at the RNA primer and then constructs a new complementary strand of DNA by adding DNA nucleotides. DNA replication is semi-conservative, meaning one strand is the template from the original DNA double helix and a new complementary one is built.
For mitosis, once the DNA is replicated, the DNA strands are condensed around histone proteins to form chromosomes. Chromosomes are made of two sister chromatids that are attached to each other at the centromere. When the chromosomes are separated during anaphase, the genes that each chromosome codes for is now found in the daughter cells. The new daughter cells are identical to the parent cell.
Over the past few years, teams of international scientists have been working to produce a synthetic genome for bacterial cells. It was only in the last few months that teams have successfully engineered the first synthetic genome for a eukaryotic cell. Using everyday brewer's yeast, scientists altered the yeast's third chromosome to create a totally synthetic version that can function normally in the yeast genome. The chromosome on the right represents the original yeast chromosome 3. Using a series of deletions, insertions, and base substitutions, scientists removed 50,000 base pairs of non-essential genes that were repetitive or jumping genes, genes that have caused mutations that were left by viruses that had invaded the cells. By taking the snippets of synthetic DNA, they created the chromosome on the left. Taking the new chromosome, now called SYN3, they attach LOX PSYM sites to SYN3 that will enable genetic scrambling. Scrambling is an acronym that stands for Synthetic Chromosome Rearrangement and Modification by LOX PSYM Mediated Evolution. LOX sites are non-directional and have no orientation they need to combine in, making it perfect to produce a whole new genome. Cre recombinase attaches to the LOX sites and actually performs the genetic scrambling. Computer programs developed by the scientists create random sequences that the genes can be combined in once the LOX sites and Cre recombinase have been activated. This process creates millions and millions of genetic variations that can be put back in the cell. It has been described as creating a new deck of cards to be able to create the best hand to play. What combination will produce the yeast cell with the highest fitness? Once the SYN3 has been perfected, it was reinserted into the yeast cell to backcross and see if the synthetic chromosome was viable.
This figure shows that the loss rate of the SYN3 DNA was not significantly different from the wild-type DNA. This means that the SYN3 yeast is viable. The fact that a synthesized DNA can be viable opens many doors in the scientific community.
Previously, the genome of a bacteria had been synthesized. However, the significance of the yeast genome being synthesized is that yeast is a eukaryotic organism. This opens the door to many advances in synthetic biology. In the future, perhaps we'll be able to synthesize other eukaryotic genomes, paving the way for advances in medicine, organ synthesis, food, and other things.